Hey gang, it's Professor Roth with another exciting episode of Biology 1 Online, and today we're going to talk about cell transports. So, question, how do cells transport molecules into and out of the cell? Well, the simple answer is there are many different ways. If it's small, if it's medium-sized, or if it's large, all could get through in different ways, and your cell has a different method for moving things in and out, depending mostly on size. But one of the ways that we can get in, and one of the simplest ways to get in, is straight up diffusion. Just good old-fashioned diffusion. So you're probably asking, well, what is diffusion then? Well, it's simply put that molecules move from a high concentration area to a low concentration area. So imagine if you're in a room and there's no air or breeze or anything blowing around the room, and you spray perfume or something into one corner. Even though the air isn't moving, there's no fans or anything, eventually the smell of the perfume would make it to the other side of the room. And that process, where it does that, from a high concentration of perfume molecules on one side, spreading out across the room until it's evenly spread, that process is diffusion. And so here's a good little example of that. So let's say we start off with the molecules over here. Now keep in mind, atoms are constantly moving. Those electrons are orbiting around, the protons are slowly chasing it, so it kind of, or it's a little wobble. And if you put them close to each other, they bump into each other, and they push on each other, and they shove each other out, and they keep doing this until they're evenly spaced. And that's what we call going from a high concentration to a low concentration. Things can do this through your cells, and it's one of the ways we can move some types of molecules and our atoms and compounds in and out of the cell. So molecules can move. Generally, they want to spread out until they are evenly spaced. Examples of salt or sugar cubes, when you put them in water, they'll simply dissolve. But what does that mean when we're dissolving? Well, that means that they're spreading out through diffusion. And as you can see here, you put a sugar clump or a salt cube or something into water, and the water is polar, so is the sugar and the salts, and it will start pulling them apart, and they bump into each other until they spread out. This is why you dump sugar in water. You can come back later, and it's more or less the entire water is going to be sugary. It'd be really weird to wake up one day and let's say you made Kool-Aid the night before, and then you wake up the next morning and all of the Kool-Aid dye and all of the sugar had pulled into a clump over on one side. That's not how things work, right? It spreads out. And that process is just good old-fashioned diffusion. So this works well for molecules dissolved in water and suspended in air. But to get through a cell, you've got to go through the cell membrane. Remember, a cell membrane is a phospholipid bilayer. It's two layers of lipids. And while some things can move through it, not everything can. And so that's going to be why we call our cell membranes semi-permeable. Yes, things can move in and out, but not everything can move in and out. So what can diffuse through a cell membrane? Well, big answer is oxygen and carbon dioxide. Things that are small can diffuse through. And again, our oxygen and carbon dioxide would all be for our respiration. It's how our cells take in the oxygen that they need and how they release and get rid of the carbon dioxide that they build up. So that's about it. Everything else is kind of too large. But there is one other little thing that can move through diffusion, and that's going to be water. But can water move through a cell membrane? Can it? Daggum straight it can. And we call that osmosis. Osmosis is a special case of diffusion specifically for water through a cell membrane. Nothing else. Water moving through a cell membrane is osmosis. So osmosis is water wants to move towards the greatest concentration of ions. Ions are anything like salts or sugars or anything else that water is able to dissolve. We just kind of call those ions. And it moves towards the greatest concentration of ions in order to dilute them. This special movement is simply called osmosis. So let's take a look at that. So here's osmosis. So let's say we have a beaker A. It's got 25% sugar in it. We've got another beaker, beaker B. It's got 75% sugar in it. And let's take a cell that's got 50% sugar. All right, so let's see where the water is going to go when we put this cell into the beaker. So if we put the cell over here, what has more sugar, the beaker or the cell? Well, the cell has a higher percent of sugar, so in osmosis, the water is going to move towards the highest concentration. So it's going to go into the cell to try to delete that sugar down. What if we take that same cell and we put it over in beaker B? Well, same principle, it's going to move towards the greatest concentration, but this time it is outside the cell. There's 75% outside, 50% inside. So the water is going to move out of the cell and try to dilute the ions that are in the solution. So, like we just said, beaker A, water moves towards the greatest concentration of ions, so water moves into the cell through osmosis. In beaker B, water moves, it still moves towards the greatest concentration of ions, but that's going to be outside, so it moves out of the cell. And again, all this is through osmosis, because this is the movement of water through a cell membrane. Now, some people think, well, why can't the sugar move? Well, sugar is too big. It won't fit through the cell membrane, not through diffusion. So it's always going to be the water that moves. All right, so now that we've talked about that a little bit, let's look at the solution types. If we took beaker A and we put the cell into it, we said that beaker A would be a hypotonic solution. Hypo meaning less than, and so it's a hypotonic solution because it has less ions than the cell. So a cell in a hypotonic solution, what do you think is going to happen to it? We said the water is going to move into it, and it's going to swell up and swell up and swell up and swell up and burst. And when things burst, we simply call that lyse or lysis. It means to cut, split, or pop. Well, what happened to the same cell in beaker B? Well, when we put it in there, we said that was going to be a hypertonic solution, meaning that the beaker B has more ions in it than the solution inside the cell. So a cell in a hypertonic solution, the water is going to move out of the cell, and what happens to the cell? Let's see. Oh, it shrivels up and it dehydrates like a raisin and gets really small. So that's kind of what happens in those two solutions. But hypotonic and hypertonic, hypo meaning less than, hyper meaning more than, greater or less than what? Hypotonic, the solution is less ions than the cell. Hypertonic, the solution has more ions than the cell. But that's not the only two conditions that it can be, right? You can also have equal to, and that's right, don't forget about isotonic. This is where we have an isotonic solution where the concentration of ions is equal. So what would happen in that case? Well, really nothing. You could say that no water moves in or out because it's not going to move through osmosis because it's equal amount of salts on either side of the cell and in the solution. But you could also say that if water does move in, it moves at the same rate out. And so it balances out. So don't forget about isotonic. So think about this for a second now. How would your cells respond when you drink these different drinks based on their solution type? Let's look at the first one. Okay, so we got a Coke. Coke's got a lot of sugar in it, Do you think it's got more sugar than the cells, like say your cheek cells? I think so too. I think it does. I think it has a lot more. So what would happen to your cheek cells when you drink Coke? Well, there's more sugar in the Coke, so the water is going to run out of your cells towards the sugar in the Coke. So your cells would dehydrate. So this would be a hypertonic solution. So what about pure water? Well, if it's just pure water, just pure H2O, and you drink it, there's more salt and ions in your cells, and so the water's going to run into your cell, and your cell's going to swell up and pop. And so we call that a hypotonic solution because the cell has more salts and ions, sugars in it than the water does. But what about this guy? You gotta stop for a second and think about this one. Now, back in the day, like in the 60s and 70s, when this stuff was invented, and it's called Gatorade for a reason. It's a sports drink that was made to aid the Florida Gators, the football team, to help them stay hydrated and perform better. Back in the day, it wasn't so sweet as it is now. It's become more of a commercialized drink, and they pump lots of sugar into it to make you buy it, so you know it tastes good. But really, it used to not have as much salts and sugars. It was isotonic to your body. That's why it worked so well. Your cells didn't shrivel up. Their cells didn't pop. They stayed perfectly fine, and that's how we wanted them to stay. If you're going to transport like something like a liver across country so it can go through a transplant, which type of solution would you put it in? Would you put it in a hypotonic, an isotonic, or a hypertonic solution? Well, hopefully you'd be smart enough to put it in a isotonic solution, so that way the cells don't degrade, they don't pop, shrivel, or anything, so that way it stays nice and healthy until it gets to its wherever it's supposed to go. So which juice you drinking? Why? Well, we just answered that. So yay, good job, you guys. You're doing great on this isotonic, hypertonic, and hypertonic solutions. So let's jump over to the next section.